I'm excited to introduce our next speaker. Emma West is the co-founder and CEO of Digital Bio. Going to tell you about one more exciting platform. Take it away, Emma.
Okay, hi everyone. My name is Emma West, co-founder and CEO of Digital Bio, and yeah, I'm super excited to tell you guys a story that we really haven't shared broadly yet, but about function-first biologics engineering in the era of genetic agency. So this is me. Great. So we work on solid tumors at Digital Bio, and maybe I can just get a show of hands for how many of us have loved someone who's been affected by cancer. It's everyone. And you know, the thing that I've loved about today is I've spent the last few months preparing for investor conversations and partner conversations, and I'm talking about cancer as an indication and a market. And every conversation I've been in today has been a sobering reminder of the fact that this is so deeply personal for all of us. So thank you, Eric, for bringing together so many amazing people on such an important mission. It's truly such an honor to be here alongside so many of my idols that I've been looking to for many years. So anyway, we are Digital Bio. We work in solid tumors. The first thing that you notice when you look at any solid tumor is that they are wildly unique. And the reality is that every human tumor is a completely unique disease. It is a continuously evolving cellular system in the context of an already uniquely evolved human. And that makes it really, really hard. And like what Martin said, where do you even start when you have so many layers of genetic dysregulation, RNA dysregulation, protein dysregulation, all this tissue gunk that has built up? And it makes it really hard to have genetic agency. And we've really pushed the limits of targeted cancer therapies from a genetic level and seen incredible success. But at Digital Bio, we're really asking ourselves, what tools do we have to go beyond where we can get with simple genetic interventions to tune therapies for individual patient biology when genetic information isn't simple to pin down? And there's some really remarkable data around how predictive testing drugs in tissues can be to predict whether a patient will respond to a particular medicine. And I think there's a couple of reasons for this. There's just the ability to test a drug on the backdrop of patient biology is obviously an advantage. But there's also just a sheer set of complex translational functions that are completely intractable to assess in any other system. Like what Martin said, you need multiple cells to be interacting to even see how these systems are responding to a therapy. And we're very excited about this. And there's been a lot of work in the very late stages of clinical deployment for how do you use tissue organoids to position therapies in the right patients. But what we're asking ourselves is, what would it take to use testing medicines in tissues as a design parameter in the earliest stages of discovery? And we saw some really nice work from Pierce and Manifold about this. But the reality is functional testing in tissues is hard. It's hard in vivo, it's hard in organoids. It's very hard to scale. And that's not just a biology problem of understanding what organoids to build. Of course, that's part of it. But it requires breakthroughs across measuring patient biology, what are the important features to recapitulate, engineering these tissues at massive scale, and being able to engineer drugs that can intervene across multiple different pathways to elicit the combination of responses required. It also requires innovations in how do you actually assess whether the drug is working and what it's doing with high resolution. And this is what we've been very focused on building. We actually started the company based on a new spatial sequencing technology that allows you to unify high-res imaging and next-gen sequencing. And everything I'm going to show you today, we've built in the last 10 months to go from having that technology to showing how you can leverage spatial functional measurements to design, screen, and build medicines. So we've also been working on building scalable 3D tumor organoids so that we can test individual antibody medicines in the context of tissues. We've built out capabilities around multi-targeting protein drugs and really being able to rapidly generate libraries and expose them in a controlled way to these microenvironments. And then again, we use that same spatial sequencing technology to read out what the heck is happening when you put that in the context of an organoid. So yeah, this is our platform. Our goals are to be able to really design, select, optimize drugs based on real patient biology. And what I'll tell you today is a far cry from this promise of being able to take a biopsy from a patient and test a drug in it. So I'll remind you of that several times so you don't think we're doing something much cooler that we're planning to do in the future. But our goal is to be able to test on the order of 1 million drugs in tissue models per year. And this opens many, many avenues across how do we actually train models that can predict tissue level functions rather than things like binding and solubility. And to be able to expand across any tissue level function. And yeah, every hit coming out of our platform, we aim to have built-in mechanistic validation. So how does it work? We start from targets and a translational mechanism that we are aiming for. We then source binders and turn those into multispecific antibody libraries. So these are combinatorial. They can leverage different types of binders like VHHs, SCFEs, et cetera. Or they can be residue tuned for additional functionality. We then go through a series of cell-based screens and organoid screens where we can do all of the standard measurements around affinity, purity, et cetera. But in the organoids, we can look for things like real tissue level responses. And all of this comes together to be able to treat biologics discovery as a multi-parameter optimization problem. We know in biologics discovery, if you're tuning affinity, that can be directly in contest with other very important properties. And this allows us to, for the same high throughput library, co-optimize for these really important functions. And as a company, we have goals of creating spatially intelligent medicines. To us, this means they have the ability to sense and process multiple signals in a tissue microenvironment that allows them to respond accordingly. So why is this happening now, and what are we building on? Binder generation is easier these days. You have AI design, which is pretty amazing, and you can also run display screens quite readily. Multispecific antibodies, we can do those. Pharma and CROs can really brute force a lot in cell-based assays, but there's a real barrier in these high-resolution tissue-based readouts. So I'll talk a little bit about the organoid screening. So the way that this works is we create 1 million of these little tumor microenvironments. And as I mentioned, these are currently not patient-derived. They are patient-immortalized tumor models. We then can expose each one to an individual multi-targeting biologic. Here we're actually detecting that drug with an antibody. We then can co-culture with patient-derived immune cells and freeze those interactions in time and do high-resolution imaging to read out what's happening. And then we use robotic microscopes and AI to actually scan through and find the ones that have optimal responses. And then we can read out with our spatial sequencing technology which drug was there. So I'll just show you a really quick example. We took a combinatorial library of 5,000 T-cell engagers. We tested them in these cell-based assays and organoid screens, and I'll show you a couple of results. So this is what it looks like after we generate these organoids. We condense them so that we can actually go through and scan them rapidly with a microscope. This is what we call a pate, which is kind of gross. But each of these is a single experiment where each one has been exposed to an individual drug. And then this is a little close-up. Here we're actually looking at, is the drug being produced? Is it stable? Is it localizing to the surface? You can see an example of one where that is not true and one where it is. And then we use the spatial sequencing after we categorize each model to read out which drugs are there. We can look at many different things. So this is the drug production and localization. We can see different levels of things like antigen binding. We can see tumor killing. And we can see even immune cell infiltration. And we're currently working on these co-culture systems and optimizing this to be able to tease apart not just our immune cells killing the tumor, but what are the actual high-resolution states they're assuming. And then, yeah, this is just some of the data that came out of this screen across a couple of different parameters, binding and killing. We're able to do this for on the order of 5,000 engagers today. And then just a summary of what happens when we take some of those hits from that screen and move them into full trispecific drugs and evaluate the function of those drugs in the context of cell and tissue models. So this is some of the data from a cell-based assay. What we found here is one of the hits that we've identified in the screen. When we put it into the trispecific, we're seeing more than 20x dual target gated activity. The goal here was take 2 clinically validated targets that have fallen short in the clinic and in terms of potency on tumor, and increase the potency in the context of both targets. So we're quite excited about this molecule. We're moving it in vivo soon, but we're very excited to continue to turn the crank on this platform. This was our very first screen that we run. Okay, and a couple things to close. A sober reminder that today we are not doing this yet in full patient-derived organoid models, but we are using representative, simple tumor models looking across thousands of variants for multiple functions. And in the future, we have a vision of expanding this to be able to really explore the patient model space and generate a set of patient representative models that span the biological diversity that we observe in these tumor indications. Also expanding the number of different functions that we can probe for in a single screen so that we're really able to optimize for translational properties. And yeah, the last thing is that we really dream of this future where a patient walks into the clinic and there's an arsenal of targeted medicines that could be rapidly fine-tuned for their biology, and where you're actually integrating bedside functional drug testing to say, will a patient respond to which combination of therapies? And this really will come at the intersection of scalable measurements, better patient models, and drugs that have the ability to be rapidly tuned for individual patient biology. And yeah, we're working on multispecifics, but these are in many ways the scaffold of like every other biological modality as shuttles or as sensors and switches. And we're very excited about this future of integrating the incredible biology that has evolved in nature with really innovative engineering, as many of you have paved the way for. So that's us. And this is my amazing team. Yeah, really grateful to be here. We're a team of 13 people, and they work very, very hard, so I had to give them a shout out. Thank you.